This image is kind of nice. Um, what it does is slice the eye um, horizontally. Right, so we're looking down into the eye. What it's showing us is the white portion, the sclera and the cornea, and it's showing us the choroid, which is kind of a pinkish red color, and in um, deep to that is a yellow layer. That's going to be our retina, which we're going to talk about next. But you can see our middle layer or vascular tunic is going to be made up of the choroid, the ciliary body, and you'll see that kind of ovally white lump there. That's the lens, that's representing the lens, and you'll see those strands holding it in place. Those are the suspensory ligaments that we talk about in lab. Now, last thing we talked about was the iris, which is the colored part of your eye, and the pupil. And the iris's job is to control how large or small that opening is, depending on how much available light, how much ambient light, as we say, there is. If there's a lot of light, the pupil is going to be choked down. It's going to be much smaller because we don't need as much light coming in. But if it's dark out, the pupil is wide open to get in as much light as possible. Now, our next layer is our nervous tunic, or the retina. The retina is easy. The retina is only the retina. For the fibrous tunic, we had the sclera, we had the cornea. For the vascular tunic, we had the ciliary body, the choroid. There's other pieces. For the nervous tunic, it is synonymous with, it is the same as the retina. It lines only the posterior portion of the eye because that's where the light is going to come in through the pupil and ideally hit the neurons on that posterior portion of the eye. We're going to be able to find 3 layers of neurons that make up our retina. We have the photoreceptor layer. Now, what do you think the job of the photoreceptor layer is? To receive light. All right, so they're going to be our receptor cells that we talked about in a few lectures ago. Next, we have the bipolar cell layer. This bipolar cell layer is referencing the structural classification of these neurons, and the ganglion cell layer. These neurons are going to be arranged one after the other. So the photoreceptor layer is going to be stimulated by the light coming into the eye. It's going to send a signal to the bipolar cell layer, which is going to send a signal to the ganglion cell layer. And you see here on the right side, those cells lined up one right after the other. The rod and the cone are the photoreceptor cells. Then we have our bipolar cells, and then we have our ganglion cells. Talking about our photoreceptor layer, these are the visual receptors that are going to be stimulated by light rays. So the light coming into your eye through the pupil, it's going to go through the cornea, into the pupil, into the back of your eye. The photoreceptor layer of neurons will be stimulated by that light. There are 2 types of photoreceptor neurons. First are the rods. Now, again, sometimes scientists are nice, makes life nice and easy for you. This is one of those instances. The rods are called rods because they're shaped like a rod. The cones are called cones because they are, you got it, shaped like cones. So that makes life a little bit easier for us. These rods are specialized for vision in dim light. So you get up in the middle of the night to go to the bathroom, right? You have one little light at the end of the hallway. Your rods are in charge of vision at that point. They allow for discrimination of shades between light and dark. And if you think about it, right, you're sitting in your room and you're just kind of looking around. You woke up, you heard something in the middle of the night. It's hard to determine color or shape, right? You're really just kind of getting broad strokes of what's inside of your room. These rods allow your eye to discriminate shape and movement. These are what causes children to have nightmares or to see weird things in your room. If you think about it, if you're kind of waking up and you're looking around your room going, what the heck is that? It might look like somebody's sitting in a chair in your room, or there's an animal in the room with you. You turn on the light and it's a sweatshirt. But because of the dim light conditions, you're only using your rods, and they're just kind of giving you basic shape and distinction of what's in the room with you or in the area with you. Rods contain a light-sensitive pigment called rhodopsin, and that's what reacts to the light. That picks up the stimulus of the light, causing the action potential to begin in that neuron. This is a electron microscope photograph, of the rods, and you see that they are in fact rod-shaped. They're longer than they are wide. Next we have our cones. They are specialized for color vision and visual acuity. You might have been thinking, well, that's okay, great. You got these rods and they give you a general sense of what's going on, but why can I see color? I can see color. I can see very clear, distinct lines when I turn lights on. Okay? That's because your cones are light sensitive. They need to have light to stimulate them. Now, if you look at this photograph, you're going to see that you have rods in it as well as cones, and they are kind of the shorter, fatter guys because they are cone-shaped, right? But again, these are the cells that are responsible for you being able to see color and to be able to see with great clarity. Visual acuity means to see those sharp, clear images that you typically are used to seeing. These cones contain light-sensitive pigments, and there are 3 of them. Erythrolabe is the pigment that allows you to see shades of red. Chlorolabe allows you to see green, and cyanolabe allows you to see blues. Your eye contains about 6 million cones and 120 million rods. Question I like to ask is, if cones are responsible for visual acuity, why don't we have many, many, many more of those instead of so many more rods? Well, it comes down to their basic job. Cones are stimulated by light, and when there's light, you don't need as many of them. Whereas with our rods, they are functioning in dim light, and we want as many as possible to be activated, to be stimulated by any small amount of light that does in fact enter the eye. The cones are concentrated in something called the central fovea within the macula lutea. The macula lutea is the de facto center of the retina, and the central fovea is the bullseye, if you will, of the macula lutea. So this is where your vision is going to be most acute. This is where this will allow for the sharpest vision. That is, if the light is going to be directed through the cornea, through the pupil, through the lens, and focused directly on that sweet spot, that macula, because that's where our cones are most concentrated. And that's what happens when we put a lens in front of our eyeball, whether it's a contact lens or eyeglass lens. It's bending the light, it's refracting the light, so it's shifting where that light's going to hit in your retina. So we're moving it so it's going to hit directly on that macula to give us the sharpest vision possible. And again, this is just another diagram of showing you the macula lutea is the larger circle, the central fovea is the bullseye of that, where we're going to have the highest concentration of rods. This is a photograph, a simulation of someone with macular degeneration. Because the macula has got the highest concentration of these cones that will allow for the sharpest vision. If that area breaks down or degrades, this is what's left. Individuals that have macular degeneration oftentimes are just left with peripheral vision. right? This is a good example. Someone with normal vision, you're seeing these two little boys on the right. Someone with age-related macular degeneration, you're seeing this, what you're seeing is represented by the image on the right. The edges you can see somewhat clearly, but the center portion of your field of view is going to be destroyed because those neurons are dying off in the central fovea. The lens is a transparent structure, right? And again, it makes sense that it's going to be transparent if it's towards the anterior portion of the eye and we want the light to go to the posterior, inside posterior part of our eye. They have to be transparent. We have to be able to allow the light to pass through. So the lens allows that light to be focused onto the retina, theoretically focusing that light directly onto the central fovea of the macula. The lens is going to be held in place by those suspensory ligaments that we talked about, and the suspensory ligaments are held in place by the ciliary body. Now, what we're going to talk about is the events of vision, that extra sheet that I indicated you needed to print. And we're just going to talk our way through looking at these diagrams, all right, kind of superimposing what we're seeing on that handout with what we're seeing on our screen. And again, these are going to be the diagrams that I had suggested you might want to either look at or print off for yourself. Now, this isn't a great diagram. I keep it because, as you can see, the names of the muscles are labeled in, or the names of the structures of the eye are labeled in Arabic, and that was the best diagram for years that I could find. I have since found a much better diagram, okay? So, bottom line is, we want to know, how does light enter the eye? How does light entering the eye trigger vision? Because of this is a step-by-step process, I'll stop here so that we can get the whole events of vision in one lecture.